Hello guys, today we will talk about major histocompatibility complex molecule and antigen presentation to T lymphocyte. Dedication to spirit of my mother. The principal function of T lymphocyte are to eradicate infections by intercellular microbe and to activate other cells such as macrophage and B lymphocytes. To serve these functions, T cells have to overcome several challenges. Firstly, there are few naive T cells specific for any antigen. Number two, the function of most T lymphocytes require that they interact with other cells like dendritic cell, macrophage, B lymphocyte. Different T cells have to be able to respond to microbial antigen in different cellular compartment. Probabilities of antigen recognized by T lymphocytes. The early experiment showed that the physicochemical forms of antigens that are recognized by T cells are different from those recognized by B lymphocytes and antibodies. And this knowledge led to the discovery of how antigens are seen by T cells. Several features of antigen recognition are unique to T lymphocytes. Number one, most T lymphocytes recognize only short peptides, whereas B cells can recognize peptides, proteins, nucleic acid, carbohydrates, lipids, and small chemicals. T cells are specific for small chemicals, substances such as dinitrophenol, urushiol of poison ivy, beta-lactams of penicillin antibiotics, and even nickel ions. In this situation, it is likely that the chemicals called haptens bind to sulfur proteins, including MHC molecules, and that T cells recognize the hapten conjugated peptides or altered MHC molecules. The antigen receptors of CD4 and CD8 T cells are specific for peptide antigens that are displayed by MHC molecules. As we will see in chapter 8, MHC recognition is also required for the maturation of CD4 and CD8 T cells. And this ensures that all mature T cells are restricted to recognizing only MHC molecule with bound antigens. MHC molecule can bind and display peptides and no other chemical structure. And this is why the majority of T cells recognize only peptides. As we will discuss later, MHC molecules are highly polymorphic, and variation in MHC molecules among individuals influence both peptide binding and T cell recognition. A single T cell can recognize a specific peptide displayed by only one of the large number of different MHC molecules that exist. This phenomenon is called MHC restriction. The feature of MHC dependent antigen recognition by T lymphocytes. Feature of antigen recognition by T cells. Most T cells recognize peptide and no other molecules, and that due to only peptide bind to MHC molecule. T cells recognize linear peptide and not conformational determinant of protein antigens, and the explanation for that is linear peptide bind to cleft of MHC molecule and protein conformation is lost during the generation of these peptides. T cells recognize cell-associated and not soluble antigens. Due to most T cells, receptors recognize only peptide MHC complex, and MHC molecules are membrane protein that display stably bound peptide on cell surface. CD4 and CD8 T cells preferentially recognize antigens sampled from the extracellular and cytosolic pool respectively. Pathways of assembly of MHC molecule ensure that class II molecule display peptides that are derived from extracellular proteins and taken up into vessels in antigen-presenting cells, and that class I molecule present peptide from cytosolic proteins. CD4 and CD8 bind to non-polymorphic region of class II and class I MHC molecule respectively. Antigen capture and the function of antigen presenting cells. Cell culture experiments showed that purified CD4 T cells should not respond to protein antigen, but they respond very well if non-T cells such as dendritic cell or macrophage were added to the culture. These cells called antigen-presenting cells. By convention, antigen-presenting cells is still the term used to refer to specialized cells that display antigen to CD4 T lymphocytes. As we will see later in this chapter, all nucleated cells can display protein antigen to CD8 T lymphocytes, but they are not all called antigen-presenting cells. Different cell types function as antigen-presenting cells to activate naive T cells or previously differentiated effector T cells. Dendritic cells are the most effective antigen-presenting cells for naive T cells, and therefore for initiating T cell response. And macrophage and B lymphocytes also function as antigen-presenting cells, both mostly for previously activated CD4 helper T cells, rather than naive T cells. Dendritic cells, macrophage, and B lymphocytes express MHC class 2 and other molecules involved in stimulating T cells, and are therefore capable of activating CD4 T lymphocytes. For this reason, these three cell types have been called professional antigen-presenting cells. However, this term is sometimes used to refer only to dendritic cells because this is the only cell type whose major function is to capture and present antigen, and the only antigen-presenting cells capable of initiating primary T cell response. Antigen-presenting cells display peptide MHC complexes for recognition by T cells and also provide additional stimuli that are required for the full response of the T cells. Since antigen is the first signal, these additional stimuli are sometimes called second signal. They are more important for activation of naive T cells than for re-stimulation of previously activated effector and memory cells. The membrane-bound molecules of antigen-presenting cells that serve to activate T cells are called co-stimulators because they function together with antigen to stimulate T cells. Antigen-presenting cells also secrete cytokines that play a critical role in T cell differentiation into effector cells. The antigen-presenting function of antigen-presenting cells is enhanced by exposure to microbial products. This is one reason that the immune system responds better to microbes than to harmless non-microbial substances. Dendritic cells and macrophage express toll-like receptors and other microbial sensors that respond to microbes by increasing the expression of MHC molecule and co-stimulators, by improving the efficiency of antigen presentation, and by activating the antigen-presenting cells to produce cytokines, all of which stimulate T cell responses. In addition, dendritic cells that are activated by microbes express cytokines receptors that stimulate their migration to sites where T cells are present. The induction of optimal T cell response to purified protein antigens requires that the antigen be administered with substance called adjuvants. Adjuvants either are products of microbe, such as killed microbacteria, or they mimic microbes and enhance the expression of co-stimulator and cytokine, as well as the antigen-presenting function of antigen-presenting cells. Antigen-presenting cells that present antigen to T cell also receive signal from these lymphocytes that enhance their antigen-presenting function. In particular, CD4 T cells that are activated by antigen recognition and co-stimulators express surface molecule, notably one called CD40 ligand. Its exact name is CD154 that bind to CD40 on dendritic cells and macrophage. And the T cells secrete cytokine such as interferon gamma that bind to the receptor on these antigen-presenting cells. The combination of CD40 signal and cytokine activates the antigen-presenting cells, resulting in increased ability to process and present antigens. Increased expression of co-stimulators and secretion of cytokines that activate the T cells. This bidirectional interaction between antigen-presenting cells display the antigen and T lymphocytes that recognize the antigen function as a positive feedback loop that plays an important role in maximizing the immune response. Here is a model for T cell recognition of a peptide MHC complex. As we can see here, the MHC molecule bind and display a peptide and T cell receptor recognizing two polymorphic region of the MHC molecule and one residue of the peptide. Here is the representation of different antigen presenting cells. One of the most effective antigen presenting cells for naive T lymphocytes is dendritic cell. Here is dendritic cell present the antigen to CD4, and the co-stimulation from dendritic cells is by B7 molecule, which is interact to CD28. This bidirectional interaction return the naive T cell into effector T cell. Here is an effector T cell response for activation of macrophage to kill microbe, and this activity is called cell-mediated immunity. And here is the effector T cell response to B cell in order to return the B cell to antibody producing plasma cell, and this is an humoral immunity. Here is a table of properties and function of antigen presenting cells. The cell type here is one of them is dendritic cell. Then the class 2 execution is constitutive increase with maturation increased by interferon gamma. Co-stimulator is constitutive expression is increased with toll-like receptor signal, interferon gamma, CD14 ligand interaction, and the principal function is initiation of T cell response to protein antigen. And this is a priming function because it enhances the naive T cell to be turned to effector cell. Macrophage, the expression of MHC is low or negative, increased by interferon gamma, and the expression is increased by toll-like receptor signal, interferon gamma, CD40 ligand interaction, and the function is effector phase of the cell-mediated immunoresponse. The B lymphocyte, its MHC is constitutive increased by interleukin 4. Expression is increased by T cell CD40 ligand interaction, antigen receptor cross-linking, and the principal function is antigen presentation to CD4 helper T cell in humoral immune response. Vascular endothelial cell inducible by interferon gamma and constitutive in human. Low co-stimulator may be inducible. May promote activation of antigen specific T cell outside of antigen exposure. Various epithelial and mesenchymal cells inducible by interferon gamma, probably non-co-stimulator and no non-physiologic function, possible role in inflammatory disease. Role of dendritic cells in antigen capture and display. The primary response of naive T cells are initiated in the peripheral lymphoid organ, to which microbe and protein antigen are transported after being collected from their portal of entry. The common route through which foreign antigen, such as microbe, enter a host are the skin and the epithelial of the gastrointestinal and respiratory system. Antigens may be produced in any tissue that has been colonized or infected by a microbe. The skin, mucosal epithelial, and parenchymal organ contain numerous lymphatic capillaries that drain lymph from these sites and into the regional lymph node. Some antigens are transported in the lymph by antigen-presenting cells, primarily dendritic cells, that capture the antigen and enter lymphatic vessel, and other antigen enter the lymphatics in cell-free form. Thus, the lymph contains a sampling of all the soluble and cell-associated antigens present in tissue. The antigens become concentrated in lymph nodes, which are interposed along lymphatic vessel and acts as filters that sample the lymph before it reaches the blood. Antigens that enter the bloodstream may be similarly sampled by the spleen. Morphology and population of dendritic cells. Dendritic cells were discovered as a population of cells in the mouse spleen that had a characteristic morphology with striking membranous or spike-like projection resembling the dendrites of neurons. These cells are present in most tissue and are enriched in lymphoid organ and in interface with the external environment such as the skin and gastrointestinal and respiratory tract. Most dendritic cells are thought to arise from adult bone marrow precursor, with the exception of Langerhans cells in the skin, which develop from the embryonic precursor that take up residence in the skin prior to birth. The classical dendritic cells, also called conventional dendritic cells, were first identified by their morphology and ability to stimulate strong T-cell response, and are the most numerous dendritic cell subset in lymphoid organs. Most of them are derived from myeloid precursors, which migrate from the bone marrow to differentiate locally into resident dendritic cell in lymphoid and non-lymphoid tissue. In the absence of infection or inflammation, classical dendritic cells capture tissue antigen and migrate to the draining lymph node, but don't produce cytokine and membrane molecules that are required to induce effective immune response. The function of these dendritic cells may be to present self-antigen to self-reactive T cells and thereby cause inactivation or death of the cell or T cell or generate regulatory T cells. Classical dendritic cells may be divided into two major subsets, one identified by high expression of BDCA1, CD1C in human, or CD11B integrin in mice, is most potent at driving CD4 T cells response. The other subset identified by expression of BDCA3 in human or in mice, CD8 in lymphoid tissue, or CD103 integrin in peripheral tissue, is particularly efficient in the process of cross-presentation. Some dendritic cells may be derived from monocytes, especially in situation of inflammation. Plasmacytoid dendritic cells resemble plasma cell morphologically and acquire the morphology and functional properties of dendritic cell only after activation. So it developed in the bone marrow from a precursor that also give rise to classical dendritic cell and are found in the blood and in a small number in lymphoid organ. In contrast to classical dendritic cell, plasmacytoid dendritic cells are poorly phagocytic and don't sample environmental antigens. The major function of plasmacytoid dendritic cells is the secretion of large amount of type 1 interferon in response to viral infection. In viral infection, plasmacytoid dendritic cells also differentiate into cells that resemble classical dendritic cells and play a role in presenting antigens to virus-specific T cells. The route of antigen entry. Here is the three types of antigen entry route. the skin, the gastrointestinal tract, and respiratory tract. There are two routes. One of them is with lymphatic vessel, and the other is blood-borne antigen with which filtered in spleen. Antigen capture and transport by dendritic cells. Dendritic cells that are resident in epithelial and tissue capture protein antigen and transport the antigen to draining lymph node. Resting tissue resident dendritic cells, sometimes referred to as immature dendritic cells, express membrane receptors such as C-type lectin that bind microbes. Apart from receptor-mediated endocytosis and phagocytosis, dendritic cells can ingest antigen by microvenocytosis and macrovenocytosis, processes that don't involve specific recognition receptor but capture whatever may be in the fluid phase in the vicinity of the dendritic cells. The dendritic cells are activated by microbe interaction signal and by cytokines such as tumor necrosis factor produced in response to the microbes. The dendritic cells also begin to express chemokine receptor called CCR7 that is specific for two chemokines, CCL19 and CCL21, which are produced in lymphatic vessel and in the T cell zone of lymph node. Naive T cells also express CCR7, and this is why naive T cells migrate to the same region of lymph node where antigen-bearing dendritic cells are concentrated. Activation also converts the dendritic cells from cells whose primary function is to capture antigen into cells that are able to present antigen to naive T cells and to activate the lymphocyte. Antigens may also be transported to lymphoid organ in soluble form. The collection and concentration of foreign antigen in lymph node are supplemented by other anatomic adaptations that serve similar functions. The mucosal surface of the gastrointestinal and respiratory systems, in addition to being drained by lymphatic capillaries, contain a specialized collection of secondary lymphoid tissues that can directly sample the luminal content of these organs for the presence of antigenic material. The best characterized of these mucosal lymphoid organs are bare patches of the ileum and the pharyngeal tonsils. Antigen-presenting cells in the spleen monitor the bloodstream for any antigens that reach the circulation. Such antigen may reach the blood either directly from the tissue or by way of the lymph from the thoracic ducts. The antigen-presenting function of dendritic cells. This was first shown for CD4 T cells responses, but is now known to be true for CD8 T cells as well. Several properties of dendritic cells make them the most efficient antigen-presenting cells for initiating primary T cell response. Dendritic cells are strategically located at the common site of entry of microbe and foreign antigen in epithelia and in tissue that may be colonized by microbe. Number two, dendritic cells express receptors that enable them to capture and respond to microbe. Number three, dendritic cells migrate from epithelia and tissue via lymphatics preferentially into the T cell zone of lymph node. And naive T lymphocytes also migrate from the circulation into the same region of lymph node. Number four, mature dendritic cells express high level of peptide MHC complex, co-stimulator, and cytokines, all of which are needed to activate naive T lymphocytes. Dendritic cells can ingest infected cells and present antigens from these cells to CD8 T lymphocytes. Some specialized dendritic cells have the ability to ingest virus-infected cell or cellular fragments and deliver viral proteins into their cytosol, allowing them to be presented to CD8 T lymphocytes. This process is called cross-presentation or cross-priming. In this diagram, here is a light micrograph of cultured dendritic cells in the skin, and here is in the lymph node, showing an extensive projections. And here is the Langerhans cell, the dendritic cell that belonging to skin, and here is the Langerhans labeled with fluorescent dye.
and here is the dendritic cell in the lymph node. Here is the dendritic cell with red fluorescent dye and the B cell with green fluorescent dye. The major subpopulation of dendritic cells is a classical conventional dendritic cells or plasmacytoid dendritic cells. Classical dendritic cells, which is divided to major and cross-presenting classical dendritic cells. The major classical dendritic cells, its surface molecule is PDCA1, CD1C for human, and CD11C and CD11B for mice. The cross-presenting is by BDCA3, CD141, CLEC9A for human, and CD11C, CD8 in thymus, CD103 in peripheral tissue in mice. Plasmacytoid dendritic cell is its surface marker is BDCA2, CD303 in human, CD11C and CD11B low or negative, plus P220 in mice. Toll-like receptors expressed is high level of T toll-like receptor 2, 3, 4, 5, 8, and 9. In cross-presenting, the toll-like receptor 3 and toll-like receptor 11 is the expressed toll-like receptors. The plasmacytoid dendritic cell have a high level of toll-like receptor 7 and 9. The major cytokines produced in the major classical dendritic cell is interleukin 12, interleukin 23, and tumor necrosis factor, interleukin 6. The cross-presenting by interleukin 12, interleukin 23, tumor necrosis factor, and interleukin 6. The plasmacytoid dendritic cells have type 1 interferon cytokines. In the major classical dendritic cell, the major postulated function is innate immunity, source of inflammatory cytokine, and adaptive immunity capture and presentation of antigen, mostly to CD4 T cells. In cross-presenting, adaptive immunity capture and cross-presentation of antigen to CD8 T cells. Plasmacytoid dendritic cell have antiviral immunity, early innate response, priming of antiviral T cells. In this diagram, activation of dendritic cells, whatever it is from Langerhans precursor or dermal dendritic cells precursor, and migration to lymph node. In lymph node, there are an activation of naive T cell. In this diagram here, summarization of the changes during dendritic cell maturation that are important in the function of this cell. The principal function, tissue resident dendritic cell, is antigen capturing, and activated dendritic cell is antigen presentation to this cell. The expression of FC receptor, mannose receptor, is highly expression in the tissue resident dendritic cells and is low in activated dendritic cells. Expression of molecules involved in T cell activation, B7, ICAM1, interleukin 12 is less or low in tissue resident dendritic cells and highly expressed in activated dendritic cells. Class 2 MHC molecule half-life is about 10 hours in tissue resident dendritic cells and more than 100 hours in activated dendritic cells. Number of surface molecule, it's about 6/10 molecule, and it activated in the activated dendritic cell, it's about 7/6/10 molecule. Function of other antigen presenting cells. In cell-mediated immune response, macrophage present the antigen of phagocytosed microbe to effector T cells, which respond by activating the macrophage to kill the microbe. This process is central to cell-mediated immunity and delayed type hypersensitivity. CD4 T cells recognize microbial antigen being presented by the macrophage and provide signals that enhance the microbicidal activities of these macrophages. In humoral immune response, T lymphocyte internalize protein antigen and present peptide derived from this protein to helper T cells. In humoral immune response, B lymphocyte internalize protein antigen and present peptide derived from this protein to helper T cells. This antigen presenting function of B cell is essential for helper T cell dependent antibody production. All nucleated cells can present peptide derived from cytosolic protein antigen to CD8 T cytotoxic T lymphocyte. It is important that the immune system be able to recognize cytosolic antigens such as viral antigen and mutated protein in any cell type. CD8 cytotoxic T lymphocytes are the cell populations that recognize this antigen and eliminate the cells in which the antigen are produced. CD8 cytotoxic T lymphocyte may also recognize phagocytosed microbe if this microbe or their antigen escape from phagocytic vessels into the cytosol. Other cell types that express class II MHC molecule and may present antigen to T cells include endothelial and some epithelial cells. Vascular endothelial cells may present antigen to blood T cells that adhere to vessel walls, and this process may contribute to the recruitment and activation of effector T cells in cell-mediated immune response. Endothelial cells in grafts are also targeted of T cell reacting against graft antigen. Various epithelial and mesenchymal cells may express class II MHC molecule in response to cytokine interferon gamma. The physiologic significance of antigen presentation by these cells population is unclear because most of them don't express co-stimulators and are not efficient at processing proteins into MHC binding peptide. It is unclear that they contribute significantly to the majority of T cell response. Thymic epithelial cells constitutively express MHC molecule and play a critical role in presenting peptide MHC complex to maturing T cells in the thymus as part of the selection process that shapes the repertoire of T cell specificities. The major histocompatibility complex MHC. Discovery of the MHC. The mouse MHC H2 complex. The MHC was discovered from the studies of tissue transplantation such as skin exchanged between non-identical individuals are rejected, whereas the same graft between identical twins are accepted. These results showed that inherited gene must be involved in the process of tissue rejection. Investigators showed that a single genetic region is primarily responsible for rapid rejection of tissue graft, and this region was called the major histocompatibility locus, histo tissue. The particular locus that was identified in mice was linked to gene on chromosome 17 encoding a blood group antigen called antigen 2, and therefore this region was named histocompatibility 2 or simply H2. Initially, this locus was thought to contain a single gene that controlled tissue compatibility. However, occasional recombination events occurred within the H2 locus during interbreeding of different strains, indicating that it actually contains several different but closely linked genes, many of which were involved in graft rejection. The genetic region that controlled graft rejection and contained several linked genes was named the major histocompatibility complex. Although not known at the time of the initial experiment, transplant rejection is in large part a T cell mediated process, and therefore it is not surprising that there is a relationship between graft rejection and MHC genes, which encode the peptide binding MHC molecule that T cell recognize. The MHC was discovered by searching for cell surface molecule in one individual that would be recognized as foreign by another individual. Individuals who had received multiple blood transfusion and patients who had received kidney transplants contained antibodies that recognized cells from the blood or kidney donors, and that multiporous women had circulating antibodies that recognized parental cells. The proteins recognized by these antibodies were called human leukocyte antigens, HLA. Leukocyte because the antibodies were tested by binding to the leukocyte of other individuals. An antigen because the molecules were recognized by antibodies. Biochemical studies gave the satisfying result that the mouse H2 protein and the human leukocyte antigen proteins had similar basic structure. From these results came the conclusion that genes that determine the fate of grafted tissue are present in all mammalian species and are homologous to the H2 genes first identified in mice. These are called MHC genes. Other polymorphic genes that contribute to graft rejection to a lesser degree are called minor histocompatibility genes. The immune response gene. For almost 20 years after the MHC was discovered, its only documented role was in graft rejection. This was a puzzle to immunologists because transplantation is not a natural phenomenon and there was no reason that a set of genes should be preserved through evolution if the only function of the genes was to control the rejection of foreign tissue grafts. In the 16th and 17th, it was discovered that MHC genes are of fundamental importance for all immune response to protein antigen. Immunologists found that invert strain of a single species, guinea pigs or mice, differed in their ability to make antibodies against some simple synthetic polypeptides, and responsiveness was inherited as a dominant Mendelian trait. The relevant genes were called immune response gene, and they were all found to map to the MHC. We now know that immune response genes are in fact MHC genes that encode MHC molecules that differ in their ability to bind and display peptides derived from various protein antigens. Responder strains, which can mount immune response to particular polypeptide antigen, inherit MHC alleles whose products can bind peptide derived from those antigens, forming peptide MHC complex that can be recognized by helper T cells. These T cells then help B cells to produce antibodies. Non-responder strains express MHC molecules that are not capable of binding peptide derived from the polypeptide antigen, and therefore these strains cannot generate helper T cells or antibodies specific for the antigen. It was also later found that many autoimmune diseases were associated with the inheritance of particular MHC allele, firmly placing these genes at the center of the mechanism that control immune response. Such studies provided the impetus for more detailed analysis of MHC genes and proteins. Here is the phenomenon of MHC restriction. If a mouse is infected with a virus, CD8, cytotoxic T lymphocyte specific for the virus developed in the animal, these cytotoxic T lymphocytes recognize and kill virus-infected cells only if the infected cell express alleles of MHC molecules that are expressed in the animal in which the cytotoxic T lymphocytes were generated. It was shown that the cytotoxic T lymphocytes and the infected target cells must be derived from mice that share a class 1 MHC allele. Thus, the recognition of antigens by CD8 cytotoxic T lymphocyte is restricted by self class 1 MHC allele. Subsequent experiments demonstrated that responses of CD4 helper T lymphocytes to antigen are restricted by self class 2 MHC allele. Here is an experimental demonstration of the phenomena of MHC restriction of T cell lymphocytes. If a model animal is injected by LCMV virus, the LCMV specific cytotoxic T lymphocyte is being clonal expanded Then the infected target cell with LCMV is in strain A and strain B. There is two strains of animal. LCMV T cell peptide from strain A with infected cell is being lysed. Then the strain A T cell with strain A non-infected, there is no lysis. Then strain B with strain A, which is infected. The strain B is infected by LCMV. Then the T cell of the model is not responding to the strain B. The MHC genes. The MHC locus contain two types of polymorphic MHC genes, the class 1 and class 2 MHC genes, which is encode two groups of a structurally distinct but homologous protein and other non-polymorphic genes whose products are involved in antigen presentation. Class 1 and class 2 MHC genes are the most polymorphic genes present in any mammalian genome. Unexpected extent of variation among individuals called polymorphism. This is a definition of polymorphism. In the population, the total number of HLA alleles with different amino acid sequence is estimated to be over 5,000, with more than 2,500 alleles for the HLA-B locus alone. The variation in MHC molecules accounting for the polymorphism results from inheritance of distant DNA sequence are not induced by gene recombination, as they are in antigen receptors.
MHC polymorphism may have evolved because it ensures that individuals will be able to deal with the diversity of microbes, and population will be protected from the devastating loss of life from emerging infections. But the selective pressure that have preserved such a vast number of the allele in the population are not understood. MHC genes are codominantly expressed in each individual. In other words, for a given MHC gene, each individual expresses the alleles that are inherited from each of the two parents for the individual. This maximizes the number of MHC molecules available to bind peptide for presentation to T cells. Human and mouse MHC loci. In humans, the MHC is located on the short arm of chromosome 6 and occupies a large segment of DNA, extending about 3,500 kilobases. In classical genetic terms, the MHC locus extends about 4 centimeters, meaning that crossover within the MHC occur in about 4% of meiosis. The human class 1 HLA gene were first defined by serologic approaches antibody binding. There are three classes, class 1 MHC gene called HLA-A, HLA-B, HLA-C, which encode three types of class 1 MHC molecule with the same name. Class 2 MHC genes were first identified by use of assay in which T cells from one individual would be activated by cells of another individual. There are three class 2 HLA gene loci called HLA-DB, HLA-DQ, and HLA-DR. Each class 2 MHC molecule is composed of a heterodimer of alpha and beta polypeptides, and the DB, DQ, and DR loci each contain separate genes designated A or B, encoding alpha and beta chains respectively, on each copy of chromosome 6. Every individual has two HLA-DB genes called DBA1, DBB1, encoding alpha and beta chain. Two HLA-DQ alpha genes, DQA1 and 2, one HLA-DQB gene, DQB1, one HLA-DQ alpha gene, DRA1, and one or two HLA-DRB genes, DRB1 and DRB3, 4, or 5.
The nomenclature of the HLA locus takes into account the enormous polymorphism identified by serologic and molecular methods. Thus, based on modern molecular typing, individual allele may be called HLA-A0201, referring to the 01 subtype of HLA-A2, or HLA-DRB10401, referring to the 01 subtype of the DR4B1 genes, and so on. The mouse MHC located on chromosome 17 occupies about 2000 kilobases, and the genes are organized in an order slightly different from the human MHC gene. One of the mouse class 1 gene, H2K, is centrometric to the class 2 region, but the other class 1 gene are telomeric to the class 2 region. There are 3 mouse class 1 MHC genes called H2K, H2D, and H2L, encoding 3 different class 1 MHC protein K, D, and L. These genes are homologous to the human HLA-A, HLA-B, and HLA-C genes. The MHC allele of particular invert strain of mice are designated by lowercase letters. Example A, B, named for the whole set of MHC genes of the mouse strain in which they were first identified. In the parlance of mouse geneticists, the allele of the H2K gene in a strain with the K-type MHC is called K of K, whereas the allele of the H2K gene in a strain with D-type MHC is called K of D. Similar terminology is used for the H2D and H2L allele. The mice have 2 class 2 MHC loci called IA and IE, which encode the IA and IE molecule, respectively. These are located in the A and E subregion of the immunoreceptor IR region of the MHC and were discovered to be the IR genes discussed earlier. The mouse class 2 genes are homologous to human HLA-DB, DQ, and DR gene. The IA allele found in the invert mouse strain with the K of K and D of K allele is called IA of K. Similar terminology is used for the IE allele as in human. There are actually 2 different genes designated A and B in the IA and IE loci that encode the alpha and beta chain of each class 2 MHC molecules. The set of MHC alleles present on each chromosome is called an MHC haplotype. For instance, an HLA haplotype of an individual could be HLA A2, B5, DR3, and so on. All heterozygous individuals, of course, have 2 HLA haplotypes. Invert mice, being homozygous, have a single haplotype. Thus, the haplotype of H2D mouse is H2KD, IA of D, IE of D, D of D, L of D. Here is a schematic map of human and mouse MHC loci. The human HLA is class 2 MHC locus. Here is a DR, here is a DQ and DV. Here is some proteasomes genes, and class 2 MHC locus, which is encode complement proteins and some cytokines, and class 1 MHC, which is divided to it type A, C, and B. Here is the mouse H2 molecule. Here is the class 1 MHC locus, and the other main class 1 MHC locus. This is the class 2 MHC, and here is the class 3 MHC. Here is the map of human MHC. Here is the class 2 region, which is contained DQ, DR, DV, and class 3, the factors of complement and some cytokines. Here is class 1, HLA-B, HLA-A, HLA-C. Expression of MHC molecules.
Because MHC molecules are required to present antigen to T lymphocyte, the expression of this protein in a cell determines whether the origin, example, microbial antigen in that cell will be recognized by T cells. There are several important features of the expression of MHC molecule that contribute to their role in protecting individuals from diverse microbial infections. Class 1 molecules are expressed on virtually all nucleated cells, whereas Class 2 molecules are expressed only in dendritic cells, B lymphocyte, macrophage, and a few other cell types.
This pattern of MHC expression, class 1 molecule and class 2 molecule, is linked to the function of class 1 restricted and class 2 restricted T cells. As discussed earlier, class 1 restricted CD8 cytotoxic T lymphocytes kill cells infected with intercellular microbes, such as viruses as well as tumors that express tumor antigen, and any nucleated cell can harbor a virus or develop into a cancer. Thus, the expression of class 1 MHC molecule on nucleated cell provide an essential display system for viral and tumor antigen. In contrast, class 2 restricted CD4 helper T lymphocyte have a set of functions that require recognizing antigen presented by a more limited number of cell types. In particular, naive CD4 T cells need to recognize antigens that are captured and presented by dendritic cells in lymphoid organs. Differentiated CD4 helper T lymphocytes function mainly to activate or help macrophage to eliminate extracellular microbes that have been phagocytosed and to help B lymphocytes to make antibodies that also eliminate extracellular microbes. Class 2 molecules are expressed mainly on these cells type and provide a system for display of peptides derived from extracellular microbes and proteins. The expression of MHC molecule is increased by cytokines produced during both innate and adaptive immunoresponse. Although class 1 molecules are constitutively expressed on nucleated cells, their expression is increased by the interferons alpha, interferons beta, and interferons gamma. The interferons are cytokines produced during the early innate immune response to many viruses. The expression of class 2 molecule is also regulated by cytokines and other signals in different cells. Interferon gamma is the principal cytokines involved in stimulating expression of class 2 molecules in antigen-presenting cells, such as dendritic cells and macrophages. Interferon gamma may be produced by natural killer cells during innate immune response and by antigen-activated T cells during adaptive immune reaction. The ability of interferon gamma to increase class 2 molecules expression on antigen-presenting cells is an amplification mechanism in adaptive immunity. As mentioned earlier, the expression of class 2 molecules also increases in response to signals from toll-like receptors responding to microbial components, thus promoting the display of microbial antigens. Beta lymphocytes constitutively express class 2 molecules and can increase expression in response to antigen recognition and cytokines produced by helper T cells, thus enhancing antigen presentation to helper cells. Interferon gamma can also increase the expression of MHC molecules on vascular endothelial cells and other non-immune cells type. The role of these cells in antigen presentation to T lymphocyte is unclear. As mentioned earlier, some cells, such as neurons, never appear to express class 2 molecules. Following activation, human but not mouse T cells express class 2 molecule. However, no cytokines has been identified in this response and its functional significance is unknown. The rate of transcription is the major determinant of level of MHC molecule synthesis and expression on the cell surface. Cytokines enhance MHC expression by stimulating the transcription of class 1 and class 2 genes in a wide variety of cell types. These effects are mediated by the binding of cytokine-activated transcription factor to DNA sequence in the promoter region of MHC genes. Several transcription factors may be assembled and bind a protein called the class 2 transcription activator, C2TA, and the entire complex binds to the class 2 promoter and promotes efficient transcription. By keeping the complex of transcription factors together, class 2 transcription activator functions as a master regulator of class 2 gene expression. Mutations in several of these transcription factors have been identified as the case of human immunodeficiency disease associated with defective expression of MHC molecule. The best studies of this disorder is Bar lymphocyte syndrome. Knockout mice lacking class 2 transcription factor also show reduced or absent class 2 expression on dendritic cells and B lymphocytes. Knockout mice lacking class 2 transcription factor A also show reduced or absent class 2 expression on dendritic cell and B lymphocyte, and an inability of interferon gamma to induce class 2 on all cell types. The expression of many of the proteins involved in antigen processing and presentation is coordinately regulated. For instance, interferon gamma increases the transcription not only of class 1 and class 2 genes, but also of several genes whose products are required for class 1 MHC assembly and peptide display, such as genes encoding the TAP transporter and some of the subunits of proteasomes discussed later in this chapter. This is a schematic representation of enhancement of class 2 MHC expression by interferon gamma. The interferon gamma produced by natural killer cells and other cell types during innate immunoreaction to microbes or by T cells during adaptive immunoreaction stimulates class 2. Here, it stimulates the class 2 MHC expression on antigen presenting cells and thus enhances the activation of CD4 T cells. Here, interferon gamma and type 1 interferon have a similar effects on the expression of class 1 MHC molecule and the activation of CD8 T cells. The MHC molecules share certain structural characteristics that are critical for their role in peptide display and antigen recognition by T lymphocytes. One of the shareable characteristics is each molecule consists of an extracellular peptide binding cleft, followed by immunoglobulin-like domain and transmembrane and cytoplasmic domain. The class 1 molecules are composed of one polypeptide chain encoded in the MHC and the second non-MHC encoded chain, whereas class 2 molecules are made up of two MHC encoded polypeptide chains. Despite this difference, the overall three-dimensional structure of class 1 and class 2 molecules are similar. The polymorphic amino acid residues of MHC molecule are located in and adjacent to the peptide binding cleft. This cleft, also called groove, is formed by the following of the amino termini of the MHC encoded protein and is composed of paired alpha helix, forming the two walls of the cleft, resting on a floor made up of an 8-stranded beta-pleated sheets. The polymorphic residues, which are the amino acids that vary among different MHC alleles, are located in the floor and walls of this cleft. This portion of MHC molecule bind peptide for display to T cells, and the antigen receptor of T cells interact with the displayed peptide and also with alpha helix of the MHC molecule. Because of amino acid variability in this region, different MHC molecules bind and display different peptides and are recognized specifically by the antigen receptor of different T cells. The non-polymorphic immunoglobulin-like domain of MHC molecule contain binding site for the T cell molecule CD4 and CD8. CD4 and CD8 are expressed on the distinct subpopulation of mature T lymphocyte and participate together with antigen receptor in the recognition of antigen. That is, the CD4 and CD8 are T cell co-receptors. CD4 binds selectively to class 2 MHC molecule and CD8 bind to class 1 molecule. This is why CD4 helper T cells recognize class 2 MHC molecule displayed peptides, whereas CD8 T cells recognize class 1 MHC molecule with bound peptide. Stated differentially, CD4 T cells are class 2 MHC restricted and CD8 T cells are class 1 MHC restricted because binding site for CD4 and CD8 are on class 2 and class 1 MHC molecule respectively. The feature of class 1 and class 2 MHC molecules. The polypeptide chain of alpha 1 MHC is alpha and beta 2 microglobulin. In class 2 MHC, the peptide chain is alpha and beta. The location of polymorphic residues in class 1 is in alpha 1 and alpha 2 domain. In class 2, it is in alpha 1 and beta 1 domain. Binding site for T cell receptor is in CD8, binding mainly to the alpha 3 domain. In class 2, it is on the pocket created by portion of alpha 2 and beta 2 microglobulin. Size of peptide binding cleft accommodate peptide of 8 to 11 residues and 10 to 30 residues or more in class 2. Nomenclature in human of class 1 MHC is HLA-A, HLA-B, HLA-C. In class 2, it is HLA-DR, HLA-DQ, HLA-DB. In mouse, class 1 is H2K, H2D, H2L. Class 2 in mouse is IA, IE. Class 1 MHC molecule. Class 1 molecule consists of two non-covalently linked polypeptide chain, an MHC encoded 44 to 47 kilodalton alpha chain or heavy chain, and a non-MHC encoded 12 kilodalton subunit called beta 2 microglobulin. Each alpha chain is oriented so that about 3/4 of the polypeptide to MHC molecule and be recognized by T cell. The polymorphic residues of class 1 molecule are confined to the alpha 1 and alpha 2 domain, where they contribute to variation among different class 1 alleles in peptide binding and T cell recognition. The alpha 3 segments of alpha chain fall into an immunoglobulin domain whose amino acid sequence is conserved among all class 1 molecules. This segment contains most of the binding site for CD8, but beta 2 microglobulin and a small part of the lower portion of the alpha 2 domain also contribute. At the carboxyl terminal end of alpha 3 segment is a stretch of approximately 25 hydrophobic amino acids that traverse the lipid bilayer of the plasma membrane. Immediately following these are approximately 30 residues located in the cytoplasm, which include a cluster of basic amino acids that interact with phospholipid head group of the inner leaflet of the lipid bilayer and anchor the MHC molecule in the plasma membrane. Beta 2 microglobulin, the light chain of class 1 molecule, is encoded by a gene outside the MHC and is named for its electrophoretic mobility, beta 2, size micro, and solubility globulin. Beta 2 microglobulin interact non-covalently with the alpha 3 domain of the alpha chain, like the alpha 3 segment. Beta microglobulin is structurally homologous to an immunoglobulin domain and is invariant among all class 1 molecules. The fully assembled class 1 molecule is a trimer consists of an alpha chain, beta 2 microglobulin, and a bound peptide, and stable expression of class 1 molecule on cell surface requires the presence of all three components of the trimeric complex.
The reason for this is that the interaction of the alpha chain with beta 2 microglobulin is stabilized by binding of peptide antigen to the cleft formed by the alpha 1 and alpha 2 segment, and conversely, the binding of peptide is strengthened by the interaction of beta 2 microglobulin with the alpha chain. Because peptides are needed to stabilize the MHC molecule, and unstable complexes are degraded. Only potential useful peptide-loaded MHC molecules are expressed on cell surface. Most individuals are heterozygous for MHC gene and therefore express 6 different class 1 molecules on every cell containing alpha chain encoded by the 2 inherent alleles of HLA-A, HLA-B, and HLA-C genes. Structure of a class 1 MHC molecule. The schematic diagram illustrates the difference region of MHC molecules not drawn to scale. Class 1 molecules are composed of polymorphic alpha chain. Here is the polymorphic alpha chain, non-covalently attached to beta 2 microglobulin. Here, the alpha chain is glycosylated, carbohydrated. Residues are non-shown. The ribbon diagram shows here the structure of extracellular portion of the HLA-B27 molecule with a bound peptide. Here is a bound peptide resolved by X-ray crystallography. Class 2 MHC molecule. Class 2 MHC molecule are composed of 2 non-covalently associated polypeptide chain, a 32 to 34 kilodalton alpha chain and a 29 to 32 kilodalton beta chain. Unlike class 1 molecules, the gene encoding both chain of class 2 molecules are polymorphic and present in the MHC locus. The amino terminal alpha 1 and beta 1 segment of the class 2 chain interact to form the peptide binding cleft, which is structurally similar to the cleft of class 1 molecule. 4 strands of the floor of the cleft and one of the alpha helical walls are formed by the alpha 1 segment, and the other 4 strands of the floor and the second wall are formed by the beta 1 segment. In human class 2 molecules, most of the polymorphism is in the beta chain. In class 2 molecules, the end of the peptide bind cleft are open, so that peptide of 30 residues or more can fit. The alpha 2 and beta 2 segments of class 2 molecule, like class 1 alpha 3 and beta 2 microglobulin, are folded into immunoglobulin domain and are non-polymorphic. In both chains, the transmembrane region ends with cluster of basic amino acid residues, followed by short hydrophilic cytoplasmic tail. The fully assembled class 2 molecule is a trimer consisting of 1 alpha chain and 1 beta chain and a bound antigenic peptide, and stable expression of class 2 molecule on cell surface requires the presence of all 3 components of the trimer. Humans inherit from each parent 1 DBA and 1 DBB genes, encoding respectively the alpha and beta chain of an HLA-DB molecule. 1 functional DQA and 1 DQB gene, 1 DRA and 1 or 2 functional DRB genes. Thus, each heterozygous individual inherits 6 to 8 class 2 MHC alleles, 3 or 4 from each parent, 1 set each of DB and DQ, and 1 or 2 of DR. Typically, there is not much bearing of MHC proteins from different loci, example DR alpha with DQB and so on, and each haplotypes tend to be inherited as a single unit.
However, because some haplotypes contain extra DRB loci that produce B chain that assemble with DR alpha, and some DQ alpha molecule encoded on 1 chromosome can associate with DQB molecule produced from other chromosomes. The total number of expressed class 2 molecules may be more than 8. Structure of class 2 MHC molecule. The schematic diagram illustrates the different regions of MHC molecule.
Class 2 MHC molecules are composed of polymorphic alpha chain non-covalently attached to the polymorphic beta chain. Both chains are glycosylated. Carbohydrate residues are not shown. The ribbon diagram here is showing the structure of the extracellular portion of HLA-DR1 molecule with a bound peptide resolved by X-ray crystallography. Binding of peptide to MHC molecule. Following the realization that the immunogenicity of protein depends on the ability of their peptide to be displayed by MHC molecule, considerable effort have been devoted to elucidating the molecular basis of peptide MHC interaction and the characteristics of peptides that allow them to bind to MHC molecule. Using methods such as equilibrium dialysis and gel filtration, X-ray crystallographic analysis of peptide MHC complexes has provided definitive information about how peptides sit in the cleft of MHC molecule and about the residues of each that participate in this binding. Characteristics of peptide MHC interactions. MHC molecules show a broad specificity for peptide binding in contrast to the fine specificity of antigen recognition by the antigen receptor of lymphocyte. In other words, a single MHC allele, example HLA-A2, can present any one of many different peptides to T cell, but a single T cell will recognize only one of these many possible HLA-A2 peptide complex. Each class 1 or class 2 MHC molecule has a single peptide binding cleft that binds one peptide at a time, but each MHC molecule can bind many different peptides. One of the earliest lines of evidence supporting this conclusion was the experimental result that different peptides that bind to the same MHC molecule can competitively inhibit one another's presentation, implying that there is only a single peptide binding cleft in every MHC molecule. The solution of the crystal structure of class 1 and class 2 MHC molecule confirms the presence of a single peptide binding cleft in these molecules. It is not surprising that a single MHC molecule can bind multiple peptides because each individual contains only a few different MHC molecules, 6 class 1 and about 8 to 10 class 2 molecules in a heterozygous individual. And these must be able to present peptide from the enormous numbers of protein antigens that one is likely to encounter. The peptides that bind to MHC molecule share structural features that promote this interaction. One of these features is the size of the peptide. Class 1 molecules can accommodate peptides that are 8 to 11 residues long, and class 2 molecule bind peptides that may be 10 to 30 residues long or longer, the optimal length being 12 to 16 residues. In addition, peptides that bind to particular allelic form of an MHC molecule contain amino acid residues that allow complementary interaction between the peptide and that allelic MHC molecule. The residues of a peptide that bind to MHC molecule are distant from those that are recognized by T cells. Number 3, MHC molecules acquire their peptide cargo during their biosynthesis and assembly inside cell. Class 1 MHC molecule acquire peptide mainly from cytosolic protein, and class 2 MHC molecule from protein in intercellular vessels. Number 4, the association of peptide and MHC molecule is a saturable interaction with a very slow off rate. In a cell, several chaperones and enzymes facilitate the binding of peptide to MHC molecule. Once formed, most peptide MHC complexes are stable, and kinetic dissociation constants are indicative of long half-lives that range from hours to many days. This extraordinary slow of rate of peptide dissociation from MHC molecule ensures that after an MHC molecule has acquired a peptide, it will display the peptide long enough to maximize the chance that a particular T cell will find the peptide it can recognize and initiate a response. very small number of peptide MHC complex are capable of activating specific T lymphocytes. Because antigen-presenting cells continuously presents peptide derived from all the protein that encounter only a very small fraction of cell surface, peptide MHC complex will contain the sample peptide. It has been estimated that as few as 100 complexes of a particular peptide with a class 2 MHC molecule on the surface of an antigen-presenting cells can initiate a specific T cell response. This represents less than 0.1% of the total number of class 1 molecule likely to be present on the surface of the antigen-presenting cells. Number 6, the MHC molecule of an individual don't discriminate between foreign peptide, example those derived from microbial protein, and peptide derived from the protein of that individual, self-antigen. Thus, MHC molecules display both self-peptide and foreign peptide, and T cells survey these displayed peptide for the presence of foreign antigen. The inability of MHC molecule to discriminate between self and foreign peptide raises two questions. First, how can a T cell recognize and be activated by an foreign antigen if normally all antigen-presenting cells are displayed mainly self-peptide MHC complex? The answer, as mentioned earlier, is that the T cells are remarkably sensitive and need to specifically recognize very few peptide MHC complex to be activated. Thus, a newly introduced antigens may be processed into peptide that load enough MHC molecule of antigen-presenting cells to activate T cells specific for that antigen. Even so, most of the MHC molecules are occupied with self-peptide. Also, microbes, the natural source of most foreign antigen, increase the efficiency of antigen presentation and include the expression of second signal. Second, if individuals process their own proteins and present them in association with their own MHC molecule, why do we normally not develop immune response against self-protein? The answer to this question is that self-peptide MHC complex are formed but don't induce autoimmunity because T cells specific for such complex are killed or inactivated. Therefore, T cells cannot normally respond to self-antigen.
Structural basis of peptide binding to MHC molecule. The binding of peptide to MHC molecule is a non-covalent interaction mediated by residues both in the peptide and in the cleft of the MHC molecule. As we will see later, protein antigen are proteolytically cleaved in antigen-presenting cells to generate the peptide that will be bound and displayed by MHC molecule. In the case of class 1 MHC molecule, association of a peptide with the MHC cleft depends on the binding of the positively charged amino N terminus and the negatively charged carboxyl C terminus of the peptide to the MHC molecule by electrostatic interaction. Many class 1 molecules have a hydrophobic pocket that recognize one of the following hydrophobic amino acids: valine, isoleucine, leucine, or methionine as the terminal ends of the peptide. Some class 1 molecules have a predilection for a basic residues, lysine or arginine, as the C terminus. In addition, other amino acid residues of a peptide may contain side chains that fit into the specific pocket and bind to complementary amino acid in the MHC molecule through electrostatic interaction, charge, base, salt, bridge, hydrogen bonding, or van der Waals interaction. Such residues of the peptide are called anchor residues because they contribute most of the favorable interaction of the binding. Example, anchor the peptide in the cleft of the MHC molecule. Each MHC binding peptide usually contains only one or two anchor residues, and this presumably allows greater variability in the other residues of the peptide, which are the residues that are recognized by specific T cells. In the case of some peptides binding to MHC molecule, especially class 2 molecule, specific interaction of peptide with the alpha helical side of the MHC cleft also contribute to the peptide binding by forming hydrogen bond or charge interaction. Class 2 MHC molecule accommodate larger peptides than class 1 MHC molecule. These longer peptides extend at either end beyond the floor of the cliff. Because many of the residues in and around the peptide binding cleft of MHC molecule are polymorphic, example, they differ among various MHC alleles. Different alleles favor the binding of different peptides. This is the structural basis for the function of MHC genes as immune response genes. Only individuals that express MHC alleles that can bind a particular peptide and display it to the T cells can respond to that peptide. The antigen receptor of T cells recognize both the antigenic peptide and the MHC molecule, with the peptide being responsible for the fine specificity of antigen recognition and the MHC residues accounting for the MHC restriction of the T cells. Predictably, variation in either the peptide antigen or the peptide binding cleft of the MHC molecule will alter presentation of the peptide or its recognition by T cells. In fact, one can enhance the immunogenicity of the peptide by incorporating into it residues that strengthen its binding to commonly inherited MHC molecule in a population. Because MHC molecule can bind only peptide but most antigens are large proteins, there must be ways by which these proteins are converted into peptide. The conversion is called antigen processing. Peptide binding to MHC molecule in this diagram A. The class 1 molecule shown in HLA-A2 and the class 2 molecule is HLA-DR1. The cleft of the class 1 molecule is closed here, whereas that of the class 2 molecule is open. Yes, open. As a result, class 2 molecule accommodate larger peptide than class 1 molecule. In the B shape, the side view of cut out of peptide bound to a class 2 MHC molecule shown how anchored. Here is the anchoring of the peptide to MHC class 2, holding it in the pocket in the cleft of the MHC molecule.
Processing of protein antigens. The pathway of antigen processing converts protein antigen present in the cytosol or internalized from the extracellular environment into peptides and load these peptides into MHC molecule for display to T lymphocytes. Peptide association is required for the stable assembly and surface expression of class 1 and class 2 MHC molecules. Protein antigens that are present in the cytosol, usually synthesized in the cell, generate class 1 associated peptides that are recognized by CD8 T cells, whereas antigens internalized from the extracellular environment into the vessel of antigen presenting cells usually generate peptides that are displayed by class 2 MHC molecule and recognized by CD4 T cells. The fundamental difference between the class 1 MHC and class 2 MHC pathways resides mainly in the site of peptide degradation. The difference between cytosolic and vesicular antigens has been demonstrated experimentally by analyzing the presentation of the same antigen introduced into antigen-presenting cells in different ways. If a protein antigen is produced in the cytoplasm of antigen-presenting cells as a product of a transfected gene modified so its protein product cannot enter the secretory pathway or introduced directly into the cytoplasm of the antigen-presenting cells by osmotic shock, peptides derived from the protein are presented by class 1 MHC molecule and are recognized by CD8 T cells. In contrast, if the same protein is added in soluble form to antigen-presenting cells and endocytosed into the vessel of antigen-presenting cells, peptides which may be different from the ones presented by class 1 are subsequently presented by class 2 MHC molecules and are recognized by antigen-specific CD4 T cells. The pathway of antigen processing and presentation. Protein antigen in the cytosol here are processed by proteasomes and peptides are transported into the endoplasmic reticulum
where they bind to MHC class 1. The class 2 MHC pathway, the extracellular protein antigen are endocytosed into viscera, where the antigen are processed, and the peptide bind to MHC type 2 and being displayed to CD4 T cells.